Hey, it's Mrs. Horseman again. Last time we talked about, we reviewed the difference between RNA and DNA, and we talked about the three different kinds of RNA. So just as a reminder, the three kinds of RNA that you really need to understand for this unit are messenger RNA, transfer RNA, and ribosomal RNA. Messenger RNA is going to be a longish strand, single helix that is a matching copy of the DNA strand for that gene that needs to be copied, okay? The transfer RNA, or tRNA, is responsible for bringing that amino acid to the ribosome in order for the protein to be assembled. And then rRNA is just part of the ribosome structure itself, so it's going to be the place where protein synthesis occurs. So, remember we talked there were two different parts of protein synthesis. The first part is transcription, and the second part is translation. So, let's talk about transcription. Transcription is when the messenger RNA, or mRNA, is produced by copying part of the DNA sequence onto a complementary RNA strand. So, there's a really important enzyme that does this, and that enzyme's name is RNA polymerase. RNA polymerase is going to find that one little section of DNA, or that gene, that it needs to copy, and it's going to start pulling the strands apart, okay? So back when we talked about DNA replication, we said that there was an enzyme that was responsible for pulling that DNA strand apart. This is the same idea. Before anything can be copied, those strands have to be separated. So RNA polymerase's first job is to pull those DNA strands apart, and it does that by breaking the hydrogen bonds between the DNA nucleotides. Then that RNA polymerase enzyme is going to use the DNA strand as a template, and it's going to bring in the nucleotides that are already there floating around inside of the nucleus. It's going to match those nucleotides up and make a complementary strand onto the messenger RNA strand, okay? So the messenger RNA isn't an exact copy, it's a complementary copy, which means cytosine matches with guanine. Guanine is going to match with cytosine. Adenine matches with uracil because there's no thymine in RNA, and then thymine matches with adenine. So that's what I mean by complementary to each other, okay? So the RNA polymerase is going to use that DNA strand as a template to make that complementary copy. Okay? Now, every gene, every piece of DNA has different genes on it. And we're only going to make a copy of one gene. We don't need to make a copy of the whole entire strand, okay? If I needed to give you specific information about biology, I'm not going to copy the whole entire book. I might copy a couple of pages that would be helpful in what you need, the information that you needed. So the same thing with DNA. It's only going to make a copy of one gene, that one gene that it needs to do its job, okay? Now, in order to identify where that gene starts, there's a promoter region, and that promoter region is where the RNA polymerase, it kind of bounces around until it finds it, and it's like, oh, this is where I start, and it's going to begin. That promoter region is a very specific sequence of bases that signals where that start should be, okay? There are signals in the DNA that tell the polymerase where to start copying, and that's just like the beginning of a sentence. You know when you're reading a sentence in English, the first letter is going to be capitalized, okay? Or the paragraph is going to be indented. That's a signal to you that that's where that piece of information begins. Just like a promoter region at the beginning of a DNA sequence tells the RNA polymerase, this is where the gene starts. Similarly, there's another region at the end of that DNA sequence, at the end of that gene, that's going to tell the RNA polymerase where to stop. And so if we're going to use that analogy, the English written language analogy, it would be like the period or the exclamation point or whatever punctuation at the end of the sentence that says, you're done reading this sentence, okay? So there's a section at the beginning that says, hey, this is where you start, and then there's a section at the very end that says, you're done. And that's how the RNA polymerase knows where to start and stop so it doesn't copy that whole entire strand. It's actually kind of cool that it does that. Okay, so let's talk about the genetic code. We've already talked about how proteins are made by joining amino acids into long chains. And those long chains, in case you hear this word, are called polypeptides. Poly means many, okay, and they're bonded together by something called a peptide bond. So a polypeptide is just a long chain of amino acids. And we talked about how the amino acids are those building blocks, just like Legos are the building blocks for the house that you build with the Lego structures. The amino acids are the building blocks for what becomes eventually that protein that your body is going to use. In your body right now, because of the foods that you eat, you have 20 different amino acids floating around in the cell just waiting to be used. And those 20 amino acids, I know there's only 20 of them, but when you put them together in different ways, they can make hundreds and thousands and millions of different proteins. Okay, it's kind of cool that way. So if we're going to stay with the analogy of the English language, there are 26 letters in our English alphabet. Well, there's 20 amino acids that are available in your cells right now to make up the proteins. The way that you put those letters in a sequence determines, and how many letters are in a sequence determines what that word says. Same thing with amino acids. The way that you put those amino acids into a sequence, and the amount of amino acids that are in that sequence determine what kind of protein it is, okay? So hopefully that'll help you understand a little bit that there are infinite possibilities for how these amino acids are put together to make all these proteins that your body's using right now. So we talk about the English language and the English alphabet. The language of mRNA instructions is called the genetic code, okay? That is what is in your genes right now. It makes a copy onto the mRNA, and the mRNA is the genetic code, okay? Remember, RNA has 4 different bases. It has guanine and cytosine, just like DNA did. It also has adenine, but RNA does not have the thymine nitrogen base. Instead, it has uracil, okay? So you need to make sure that you remember that. That's really important. So now that you know the mRNA is written, we've made that copy of mRNA during transcription. Now it needs to be read during translation, okay? So now we're going to look at how that genetic code is read. Just like you learned how to read the English language in books, now I'm going to teach you how to read the genetic code, which is the letters that are written on the mRNA strand. The first thing you need to know is that the genetic code is read 3 letters or bases at a time. So each of those words, each of those 3 sequence of bases is called a codon. So on an mRNA strand, every 3 bases is called a codon, okay? So for instance, you can see here, this says UCG. That is 1 codon. CAC is another codon. GGU is our 3rd codon, okay? All of those are 3 base segments. It's all in 1 long strand, so a lot of times when we see it written on paper, the nitrogen bases on paper, it might be separated by a little dash like this, just to help us keep it in order and to help us see those 3 bases, which we call a codon. Each codon signifies a different amino acid. All right, so really quick before we go into looking at how that genetic code is read and translated into a protein, I want you to stop and go back and think about these questions. What's made during transcription and why? What is the genetic code? What is the 3-letter word that represents an amino acid? What is that called? What base is found in RNA that's only found in RNA? It is not in DNA. And what are the building blocks of a protein? What makes up a protein? If you don't know those, you need to go back and look in your notes or rewatch this video to make sure that you know this information by the end of the unit. All right, moving on. There's a translation video clip that we are going to actually look at in class. In the meantime, I'm just going to briefly show you. I'm going to walk you through this process, and then we'll write down step by step what happens, okay? So we have our ribosome, and we know that the ribosome is made up of rRNA. That's our structure, okay? That is the building site. The mRNA, this strand right here, is the copy of the DNA. It's the copy of the directions, or a copy of the blueprint, okay? These are the directions for how the protein is going to be built. These are the amino acids that are put together in a chain, and the amino acids are just the tools or the supplies for the building that's going to be built, okay? So we have the directions, the instructions for how the protein is going to be built. We have the supplies to build the protein, which are the amino acids, and we also have our building site, which in this instance is the ribosome, okay? Whoops. I do want to point out to you, so I've indicated the ribosome, which is our rRNA, the directions, which are the mRNA, and now this is the tRNA. And you can see at the very bottom here, there's 1, 2, 3 nitrogen bases that are at the bottom of this tRNA molecule. Just like here, this is a different one. There's 1, 2, 3 bases, 1, 2, 3 bases. Those bases are going to pair up with the mRNA strand, and that's how it knows. It has to match up together. They're complementary to each other, and that's how it knows which amino acid is the next one in line. Sorry, it keeps jumping on me. These 3 nitrogen bases determine this is the amino acid that this specific tRNA molecule will pick up, okay? There are tons, I think 64 different combinations of nitrogen bases that you, like CGA, GCU, UAC. There's 64 different combinations of those nitrogen bases that are read in codes of 3, okay? So each one of those signifies a different amino acid, and that's how they figure out which ones to match up. Each tRNA molecule is only responsible for one kind of amino acid. So that would be like me saying this tRNA molecule is responsible for picking up a red 3-prong Lego block. And that's the only kind it can pick up. Okay, are you with me? All right, so let's see how this is made. So this is the second part. This is translation. This is reading the genetic code and taking it from the language of nucleotides that are on the DNA and RNA strand and translating it into amino acids which make up the protein. So the first step is reading the code from the mRNA. The ribosome is responsible for reading that code, for reading the directions that are on the messenger RNA strand. This is the information that tells the amino acids which, or that tells the ribosome which amino acids are next. All right, so here is our DNA strand. Through transcription, we make a copy of that onto a very similar RNA strand. So now we're going to focus on how the directions for the RNA strand actually make up a protein. Okay, remember that the mRNA was first transcribed in the nucleus, and then it was released into the cytoplasm. So it was copied in the nucleus, it went out of a nuclear pore that's in the membrane, in the nuclear membrane, and it went out to the ribosome, which is waiting in the cytoplasm. Translation actually begins when an mRNA molecule attaches to the ribosome. So the ribosome's 2 pieces, it kind of looks like a hamburger bun. So when the mRNA strand latches onto that ribosome, and then the top part of the ribosome comes down over the top of it, that's when translation begins. Every codon, so every 3 nitrogen bases of the mRNA molecule, it moves through the ribosome at a time. So the ribosome reads the mRNA strand 3 bases at a time. And remember, we call that a codon. The tRNA molecules, those are the ones that are responsible for bringing the supplies, are going to bring the correct amino acid to the ribosome, and in order to read that, the tRNA has an anticodon. It is the exact opposite of what that codon on the mRNA strand is. So the mRNA strand has a codon, the tRNA strand has the complement to that, okay, or the opposite of that. So that's going to come in and match it. When it matches, then it's allowed to release its amino acid. It's not going to give its amino acid up to that protein strand until it knows for sure by matching that it's the right one. Okay? Okay. As the mRNA then moves through the ribosome, so this one has been read, the tRNA molecule matches it, it gives up its amino acid. Now the tRNA molecule is going to go away and pick up another amino acid somewhere else in the cell. This mRNA strand is going to move through the ribosome, and now there's a new codon that's sitting in that spot, and the new codon is going to be read, and another tRNA molecule will bring in and bring its amino acid. And it's going to do this through the ribosome until it reaches the very end. There's actually a stop codon on the mRNA strand that tells the ribosome that it is done, and it allows the ribosome to then release all the pieces. Okay, that is called a stop codon. The mRNA at that point is going to, so we have this mRNA strand, the instructions have been given, we don't need that anymore. So the mRNA strand is actually going to break up into pieces and go back into the cytoplasm so it can be recycled into another type of RNA. Okay, here's a picture of that happening again. So here we go. Here is our ribosome, and this looks a little bit different than the other pictures I've showed you, but here's our DNA strand. The DNA unzips and we make a copy. Okay, that is the mRNA strand. The mRNA strand is then going to leave the nucleus and it's going to latch onto the ribosome. The ribosome is going to read that mRNA strand 3 nucleotides at a time, and we call that a codon. So here you can see this is one codon. All right, here is another codon. And this codon is being matched to its complement on this tRNA strand. This tRNA, because it matches exactly, knows what amino acid needs to be added to this chain of amino acids that's growing out of the protein. This is the next codon, so there will be another tRNA molecule that will come in and match that right there. And then it's going to add its amino acid right here onto this chain of growing amino acids. And then this whole strand of mRNA is going to move down a little bit so that now the next, So now the next codon will be available to be read and matched with the tRNA strand. And this goes on and on and on and on until it reaches the very end, or the stop signal. Okay, so let's practice. This is our little amino acid chart. So here's how you read it. When you have a codon, the first thing you have to do is figure out from our DNA strand what the mRNA strand is going to be. So here we have, ATG, GTA, GCT. All right, so we are going to make that into an mRNA strand. We are going to transcribe it and make our copy. So the copy of that would be there's no T in mRNA. UAC CAU CGA. All right, so hopefully you wrote that down. So our first codon is going to be on the mRNA strand is going to be UAC. So how am I going to figure out the tRNA? The tRNA to that is complementary, okay? So you just write the complement to it. I'm going to use that codon that I just transcribed to figure out how to use this chart right here. So we said this codon, ATG, its match or its complement is going to be UAC. So I'm going to come to this chart over here. And you guys will get a chart that looks just like this in order to be able to do our practice work. So the first thing you do is you come to the very center. You come to the very center of this chart, right here, that's going to be your first codon. So we said your first nitrogen base. So we said our first nitrogen base was U. So here we go. We're going to come into this quadrant, okay? U. We said the next one was A, so we're going to move out to the A section. And this very last ring is going to be the third nitrogen base in that. So UA, we said that C was the next one. So here's C. So the amino acid that matches with that codon is tyrosine, okay, or tyrosine. All right, so that's our first one. Our first amino acid is going to be tyrosine. The next codon we have is GTA. So we're going to match that. You should have gotten CAU. All right, so here we come over to the chart. We're going to start in the very center. C was our first nitrogen base. So we'll come to the C. We're going to look in this quadrant. We said the next one was an A, so we're going to follow this out to the next ring. Here's our A, and we said the last one was a U. So CAU, the next amino acid is histidine. All right, that's how we read this chart. Let's do the last one together. Our last nitrogen base triplet on the DNA strand is GCT. So the match of that, the complement of that would be CGA. So let's come down to our handy dandy chart. We start with the C's in the first circle. We're going to be working in this quadrant again. So C, G, we come out to the second ring, and then we follow that out to the third ring. And this tells us that the amino acid that matches that codon is arginine. All right, so that is the chain of our amino acids. This gene only happens to be 9 bases long, so our amino acid strand is only going to be 3 amino acids. That protein will only be 3 amino acids. I also want to point out, if you notice, some of these, we could get multiple combinations in order to get this amino acid. So CGG, CGA, CGC, CGU, all code for arginine. That is okay. It's supposed to be that way. We only have 4 base pairs, or 4 different nitrogen bases. And when you put that into a combination, we can get 64 different combinations of those 4 bases. That's a lot. There's only 20 amino acids. So that's like CGG and CGU are like synonyms of each other. That means it's different words that mean the same thing. So these are different codons. CGG and CGU are different codons, but they code for the same amino acid. Okay, so that's a synonym. So I just wanted to point that out to you. So now that we know how proteins are made, why the heck are they so important? Don't you think that you should know that? Probably. Proteins are often used as enzymes. An enzyme is something that just helps along reactions that are in your body. So we have talked about some of the different things that happen in your cells. Definitely the most important that we've talked about so far. Now we know protein synthesis, and there's an enzyme that helps with that. But metabolism. Okay, your body has to break down the food that you eat, and then that food that you eat is broken down into its basic parts and pushed out into the cells. Well, how the heck do you think that happens? Because our bodies make these proteins called enzymes that help those chemical reactions break down the food. And then they take those little basic pieces, so the nitrogen bases and the amino acids and the very basic fatty acids, and they put those back together into new things that our body needs in order to function the right way. So enzymes are just helpers that help all of these things happen in your cells. Super important. Your body would stop working if it didn't have enzymes, proteins, to help those things happen. Flower color. It happens, you see color in flowers because of proteins. Those proteins are pigments, and the directions for those proteins that create those pigments that make it look like a pink flower come from the DNA. Your blood type. I am type A. Okay, that is specific to me because my DNA says so. My DNA has that gene, the gene is copied, and my body makes from that gene, the copy of the gene, the proteins that go on my red blood cells that say, this is Mrs. Horseman's cell. Okay? These enzymes regulate the rate and the pattern of growth throughout an organism. So as you are all growing, it's because there are proteins that are causing that growth to happen. Proteins are everything. Your body would stop working if proteins were not present. So here's a couple questions just to think about, and we will talk about these in class as well. In a nutshell, what does translation do? You need to be able to tell me the main purpose of translation and how that works. Where does translation take place? That is very important that you know that. And I'll really think about it. If you know what translation is, you know that one of those parts of translation is locked into a specific place in the cell and it can't leave that certain place. So obviously then you would figure out where that happens, where the copy of that happens. As the codons are being read, what does each codon represent? And last but not least, why are proteins so important? You need to know the answers to these questions, and we'll talk about them in class. But if you don't know the answers to these questions, you need to go back and look at your notes and maybe rewatch this section of the video so that you do understand. You need to know there are two main steps to how a protein is made: transcription and translation. In class, we are going to do an activity that is an analogy for how baking a cake, or really following any recipe, is just like protein synthesis. We've also talked in class about how we could compare a ribosome to a job site or a building site, and then the mRNA and the tRNA are parts of that building of the protein. But really, the way to think about this isn't a play or an act, okay? Building this protein is an act in a play. In order to understand how that plays out, so how the scene happens, you need to understand there are four main characters: DNA, mRNA, tRNA, and rRNA. You need to be able to know their roles, okay, the parts that they play, before you can figure out how they all work together in this scene. All right, so I've told you 3 different ways that you can think about it, and we'll practice this more in class, but I really hope that that helps you. Honestly, if you don't understand this, go back and watch the video, ask a friend, ask me in class, because you really need to understand this in order to do well in the class. All right, I hope that helps, and I'll see you soon.